Good afternoon, dear scientists. I'd like to welcome you here. My name is Övgü Gencer. I'm the doctor at Ege University in Izmir in Turkey. During this study, I worked at IMET in USA. And today I would like to take this opportunity to talk to you about the fact of the use of different stocking volumes on the development of blue crab broodstocks. Blue crab is one of the blue swimming crab, which it comes from Arthropodophilium, Crustaceous subphylum, and Portinidae family member. On the right side, you see adults blue crabs. On the left side, the first stage blue crab larva picture. For our study, Preparaurus adult female Calnectes stapedus were collected in the Chesapeake Bay from the fall season of 2018-2019 and reared in blue crab hatchery in the Aquaculture Research Center, which we called ARC, Institute of Marine and Environmental Technology, Baltimore, Maryland, USA. Animals were kept in captivity in 5 closed circulation aquaculture system tanks, 90 centimeters weight, 110 centimeters height, 60 centimeters dimensions at 8 hours light and 16 hours dark photoperiod condition. Tanks contain 300 liters of artificial seawater with 30 ppt at 22 centigrade degrees. Zooaquatic lab monitored of water quality daily. The animals were fed daily with 10 grams or 10 percentage of their body weight of a piece of frozen squid. Female crabs carrying imminently hatching embryos previously analyzed were transferred into 50 liters artificial seawater, 30 ppt broodstock tanks. A total of 90 blue crab broodstocks have been brought to the research center, and 3 batches with 3 replications have been conducted in a total of 9 tanks. 15 broodstocks per tank in the first batch, 10 broodstocks per tank in the second batch, and 5 broodstocks per tank in a third batch have been stocked in 9 separate tanks and taken into the rearing environment. Then, the basic statistical data of the measurement values have been calculated and the relationships between them have been determined. The results regarding the effect of stock volume on growth, development, and survival have been given, as can be seen from the values in Table 1. The initial measurement values of the broodstocks in batch 2 are the highest. However, the highest final measurement values have been obtained at T3 in batch 3. In addition, 48 of the 90 blue crabs lived in and 42 of them died. The average water qualities are in the table 2. The results presented below show that the rearing of the blue crab in tanks has been carried out in 3 batches, and for 3 tanks, tanks 1 to 3, in each batch, the feeding regimens for different tanks are exactly the same. The feeding changes made over time are small. For broodstock rearing experiments, a set of stock densities 5 to 15 broodstocks per tank of broodstock survival have been shown in Figure 1. A general trend towards reducing broodstock survival rates from 100% to 23% can be seen when the stocking density is increased from 5 to 15 broodstock tanks. Similarly, a significant difference was found between these factors for the assessed intensity range. Uh, Survival rate is statistically different according to the density of stock in each of the batches. So, as the stock density increases, the survival rate decreases. The results of broodstock rearing experiments, a set of stock densities of broodstock development rates have been shown in Figure 2. A general trend towards reducing broodstock development rates from uh, 23.8 to 7.69 percentage uh, can be seen when the stocking density is increased from 5 to 15 broodstock tank. Similarly, a significant difference has been found between these factors for the assessed intensity range. The development rates are statistically different according to the stock density in each of the batches. As the stock density increases, the rate of development increases. So, in the study, the effect of stock density on development growth and the survival in blue crabroot stocks has been examined. Pearson's test has been used in the study. The results have been showed changes in broodstock rearing survival rates in three different batches. Statistical differences in survival rates have been found at higher stocking densities. These results indicate that the broodstock densities evaluated in this study indicate that increasing the density will significantly reduce survival. Similarly to our results, in the study have been observed that the survival rate of crabs increased significantly with a decrease in stock density. It has been concluded that the high stock density affects the survival rate, growth, and the development of connected Staphylococcus broodstocks. The effects of the stock density have an important place in the cultivation of connected Staphylococcus broodstocks. So broodstocks have more space than food in their cultivation at the lowest stock density. For this reason, it has shown a lot of survival and very rapid growth development. In the future studies, the optimum stock density for mass production should be determined. As studies concluded that medium density, that is the 50 larvae per liter, culture has obvious advantages and will reduce the overall cost. With its optimum stock density, it's important both for survival and growth for cost. Since cannibalism in blue crabs is lethal for crabs, it has been observed that as the number of stocks increases, the depth increase and they break each other's pressures and similar bases. In this study, we investigated how many volumes we can get to maximum yield, and it has been determined that the maximum number of stocks can be taken into aquaculture in commercial studies in the field of aquaculture. Thank you very much for your attention.